Great. Well, thanks ever so much, everyone, for coming to this Easter edition of the Plant Science Talks, and thanks very much, Sharon, for having me. It's really great to have an opportunity to speak to people about your research at the minute, when obviously a lot of us are feeling maybe a bit cut off from conferences and gatherings. So as we've said, I currently work at the University of Bristol in Imogen Sparks' group, doing a lot of sort of microscopy and cell biology, which might seem a very long way from the work that I'm about to talk about, but I have ideas about maybe how the two can connect in the future. So my broad interest in plants is very much developmental, and I'm quite interested in how cell biology processes drive some of the broader developmental phenomena that we observe. And my PhD work, which links in with this ongoing project that I'd like to present today, focused more on how the development impacts the physiology. So I feel like this makes a nice sort of pipeline from very detailed tiny things right through to how they affect the functioning of the plant. So I've called my talk Fantastic Leaves and How to Find Them. And what I'm really focusing on today is thinking about ways that we can develop leaves that do photosynthesis more efficiently, and different methods of trying to identify the traits that we need to have in those plants to maximize photosynthesis. So I've put a selection of very pretty leaves on my title slide to make it look really nice, but actually I mostly work on Arabidopsis. They're not bad leaves either, just maybe not quite as beautiful as some of these. So, oh, yeah, there we go. So the obvious first question really is, in terms of my presentation, what am I defining as a fantastic leaf? So here we have a cross-section diagram of a typical dicot leaf. I'll say that I'm not specifically obsessed with dicots. Monocots have great leaves too, for any monocot researchers joining in today. But yeah, most of my work has been on Arabidopsis, mostly because of the genetic resources available, though I have also done a bit of work in rice during my time in the Fleming lab at Sheffield. So as I'm sure most of you, if not all, are aware, we have different layers within the dicot leaf. There are the tall palisade mesophyll cells, and the more irregularly shaped spongy mesophyll cells with larger air spaces in between. There are epidermal cells at the top and bottom of the leaf that provide a barrier to regulate gas exchange and to minimize pathogen entry. And there are the stomata, which again are involved in the very specific regulation of gas exchange. So speaking of gas exchange, that is one thing that's very important to a fantastic leaf. So a fantastic leaf needs to be really, really good at taking up as much CO2 as possible to maximize photosynthesis. And it also needs to be able to balance that with loss of water, which is obviously important for transpiration, but needs to be kept in check in case of drought conditions. The other side of the coin is that a fantastic leaf needs to be really good at light capture if it's going to maximize photosynthesis. So typically these tall palisade cells are where most of the photosynthesis is thought to occur, and the chloroplasts can move around inside those cells to make sure that they're capturing as much sun as possible while minimizing photo damage. So a fantastic leaf needs to be great at catching light and CO2 and trading that off a bit with water loss. So when it comes to improving photosynthesis, obviously this is a really big topic for plant scientists. It's something we feel we need to do to counteract lots of the big challenges facing the world today. So food supply, climate change, supply of other raw materials. It's a problem that we have obviously been working on as a community for decades, and we've made some massive advances, but there probably are still areas where there are gains yet to be had. So one aspect of leaf biology that people have looked at quite a lot to try and improve photosynthesis is leaf biochemistry, whether that's photochemistry, whether that's the specific reactions involved in photosynthesis itself. people have examined different systems of photosynthesis, and people have obviously tried to engineer different photosynthetic metabolism systems into different plant species. So there's, I guess, a lot of attention given to the biochemistry of the leaf and how that could be modified to improve photosynthesis. But an area that's maybe had a bit less interest historically or attention is how leaf anatomy could influence photosynthesis. So there's a lot to unpack about leaf anatomy. The structure of a leaf, if you've spent any time looking inside them, is fantastically complicated. And that's one of the things that makes them so interesting, but also makes it very hard to get a handle on which aspects of that structure are really important for efficient gas exchange, and how those gas exchange considerations trade off with light capture considerations. So the same traits might not be optimal for both, so there may be compromises there. So yeah, there's a lot I think we still have to understand about leaf anatomy and how it forms, how it's regulated, and how it influences photosynthesis. But in the Fleming lab, where I did my PhD and where I continue to collaborate on the project I'm going to talk about today, and other bits and pieces we're still wrapping up, the sort of leaf anatomy question has been a big focus for quite a number of years now, and there have been some really interesting insights by various modes. So one thing to think about if we want to try and optimize any trait is understanding how we can access variation in that trait, because if we want to know what makes something better or worse, we need different versions to look at and see how they perform compared to one another. So just as a few examples, obviously there will be some natural variation in leaf structure. Different growth environments can give rise to different leaf structures. So both of these were aspects of variation that we looked at as part of a rice leaf modelling project, which Dr. Jen Sloan gave one of these talks about a few weeks ago. I think you can still find that on YouTube. So that model is called eLeaf, and we looked at different natural accessions of rice, and we grew rice under different CO2 conditions and looked at whether that affected the way that the plants grew. So those are some ways that you can access variation in leaf anatomy. Another one is cell growth and division. So there was a paper from the lab, I think published in 2017, where they examined a number of mutants in cell cycle regulatory genes under promoters specific to different leaf layers, and they were able to demonstrate some variation in various aspects of the leaf structure, and to show that that influenced the photosynthetic properties of the leaf. Cell separation is another really tricky one to get a handle on. This is something that I really tried to look at during my PhD, but it was really challenging to find mutants that specifically affected cell separation without having lots of other confounding effects. And that is one problem with all of these approaches. It's very hard to just specifically manipulate one very specific leaf trait, because they're also interconnected, these kind of structural properties, and many of them also relate to other properties. So for example, for cell separation, we thought the cell wall would be important, but obviously the cell wall also has lots of impacts on diffusion directly. So yeah, a complicated one, but another way of perhaps finding variation in leaf structure. And finally, another paper that came out in 2019, I think, in Nature Communications, we showed that changes in stomatal density have knock-on effects on the structure of the mesophyll. So yeah, there are lots of different ways that you can access variation in leaf structure to try and unpack how that then influences leaf photosynthesis. So another challenge for these sorts of questions is actually being able to see inside the leaf and to evaluate those structures in order to relate them onto photosynthesis. So as I said, there are lots of aspects of leaf structure. You can think about it in terms of the proportion of airspace. You can think about it in terms of the size or dimensions of those airspaces, the size and dimensions of the cells, how tortuous the pathway is for gases to move through the airspace. So there really are lots of different ways of measuring the structure of the leaf. But in order to do any of them, you have to have a clear picture of what that structure looks like. So traditional histological approaches to examining leaf anatomy are great in many ways, but they do have their limitations, such as often low throughput. It can be really hard to serially section a leaf and then reconstruct it fully in 3D. And there have been some really interesting papers over the last sort of 3 to 5 years showing that taking 2D sections of a leaf and then trying to infer 3D structure of the leaf and its properties is not a very reliable method. So it's much better, if at all possible, to image in a 3D way. So you're sectioning serially, whether that is by hand or by microscopy. So I guess the easiest, most accessible way that most of us might be able to look at a leaf in serial sections would be on a confocal microscope. So these pictures on the left are some of my confocal images of an Arabidopsis leaf, and they are very pretty, if I say it myself, but it's actually very difficult to reliably get images of this quality. There are often problems during the fixation or during the staining, which mean it's really hard to get high quality images right throughout the depth of the leaf. So an alternative approach that the Fleming Lab, amongst others, have been using for a number of years is X-ray micro-CT. So we were very lucky to have access to the Hounsfield facility at the University of Nottingham. We worked in collaboration with them to develop a method for reliably imaging leaf tissue by the micro-CT method. So I'm just going to play this little video while I explain it. So this is a 3D rendering of a micro-CT scan. So it's an X-ray based scan, just like medical CT, but obviously in very high resolution. So in a second, the green tissue will be optically stripped away in this rendering, and you'll see a yellow pattern, and that is the airspace inside the leaf. So by scanning leaves in this way, we're able to very quickly access a hugely detailed amount of quantitative information about the structure of the leaf, and to assess many of the different parameters that I've described that influence the overall leaf structure. So we published a method in 2018 to describe how we do this. So sometimes we have scanned whole leaves, as in the video, but more frequently we'll just take a small disc, as you can see here, and scan that. It's sandwiched between polystyrene to make sure that it's stable during the scanning period. And the scan is quite rapid to make sure that that leaf isn't desiccating and changing the shape and size of its cells over the period of the scan. So this final image along the top is just a picture of the X-ray scanner, so that the sample is rotated in the X-ray beams, and that allows us to detect differences in density between the cells and the air spaces. And then in the computer, we can threshold based on those density differences and ultimately extract quantitative information about the sizes of cells and air spaces in the leaf. So it's yeah, actually a very rapid and very detailed way of obtaining information about leaf structure, and we've used it quite a lot over the last few years, and I hope it's something that people will keep thinking about using, because I do think it's yeah, really useful technology. So just one example of a paper from the Fleming Lab which used this technology. So this paper was the one I mentioned before, where they had a number of mutants that varied in, so that were altered in cell cycle regulation in different layers of the leaf. And as one example of a graph here that's used the CT approach, the spongy mesophyll porosity on the x-axis was calculated from those micro CT images, and here they're relating it to carbon fixation, so that's a good proxy for photosynthesis, and you can see a correlation in this graph. So I guess often the way we address these sorts of questions, wanting to know which parameters will make a leaf good, would be to look at variation, relate it to the outcome that we want, and work out for each parameter what we're looking for. But there are some limitations here. So one of those is that we're limited in a sense in how we think about it by the range of spongy mesophyll porosities that we're able to measure. So you can see there's a sort of 10% variation within the lines examined here. But if we can't find or generate mutants with more extreme variation, then we can't ask questions about how that variation would influence more extreme cases. Another problem is, obviously this is a correlation, but it doesn't necessarily give you a mechanistic understanding of what's happening, and there are lots of follow-up experiments that you might need to do to test whether different parts of, or whether different explanatory factors are really responsible for this relationship. So to take a very different approach, the project that I've been involved in recently started in, rather than starting from data, we started by trying to build an extremely simplified model leaf, a sort of optimized geometrical model, and then later relating that to physical data and trying to see whether the model accurately predicted things that we observe in real life. So I'll explain that in a little more detail. So the first element of this simplified model, in fact, before I go any further, I should say that the modelling work here was done by a PhD student in the engineering department here at Sheffield, that's Shiv Gangapasad, and he also did lots of the reanalysis of the data that I generated in order to parameterize this model and to look at whether it was working effectively. So yeah, he's done lots of fantastic work, and it's great to be able to share this with some people. It's not currently out. So yeah, he began by considering an airspace within a leaf, and here I should say we're particularly picturing the palisade mesophyll. We haven't considered the spongy mesophyll yet in this model. And he's modelled this airspace as a tube, and he's used physical diffusion equations to consider how the length and diameter of this tube would affect the uptake of CO2. And the next aspect of this model is scaling it up from a single airspace into a 3D section of leaf. So if you imagine here, we're looking down on a leaf, and our green is cellular tissue, and the white circles represent air channels as drawn on the left. So obviously this is a great simplification, and air channels are very rarely circular, but as I'll explain later, we calculate them very much in terms of surface area, which is known to be a really important driver of CO2 uptake. So yeah, it may trouble you that this doesn't look much like a leaf, but I think in terms of building this very simplified model, this should suffice for the time being. So thinking now about how we rein in this model and know what numbers to feed into it and what numbers we expect out of it. So fundamentally what the model will do is predict the CO2 uptake for a given leaf structure. So we'll say in a given area of leaf, if our channels are this size and we have this many of them, they're spaced out in this way, how much CO2 would the leaf be able to uptake per unit time? So the micro-CT approach, which I've already described, is how we obtain the structural data to parameterize this model. I have a second slide in a moment which will help you to visualize how we converted those quite complicated looking X-ray data into some numbers that we can feed into this really simple picture. And another parameter we needed to constrain was we needed to guide the model as to the rate of CO2 uptake that it might be expecting to be able to happen for a given surface area of mesophyll. So for the same leaves that I put through the micro-CT scanner, I had first measured using an infrared gas analyzer system, the LICOR, and using the Sharkey et al. Method, we calculated a value for GM based on the relationship between intracellular CO2 and assimilation. So calculating GM can be really tricky, and I appreciate that this is probably not the most optimal method, but it gave us an opportunity to generate those data from data we already had and that were paired with these structural data. So while it's not necessarily the way that you would choose to do it if you were starting from scratch and you had the resources and facilities to conduct an entire new set of measurements, it certainly for the purposes of initially testing out the model, I think this gave us a really useful estimation of that parameter, and that guides the prediction of CO2 uptake. So as I mentioned, it seems tricky to convert these sorts of images from the CT scanner. So here black is cells and white is airspace into the diagram as I showed before. So one of the things that we know is really important for driving CO2 uptake is mesophyll surface area. So that's the surface area within the leaf that's available to take up CO2. So in terms of this single slice of leaf, that would be represented by the perimeter of the white air spaces. And the way that Shiv calculated the diameter of these channels to put into the model was based on the perimeters of the air spaces and the number of air spaces in the CT slice. So while some aspects of this structure obviously can't be represented in this much simplified structure, critically SMES, the mesophyll surface area, is equivalent between the two. So that's sort of how those parameters were put into the model in a way that ensures that some of the most critical parameters are common between the simplified structure and the true structure of the leaf. So one of the first things that we can do with this model is to ask, if we vary air channel diameter, what will the CO2 uptake be? That's sort of our major output, as I displayed in the previous, the slide before last. And one of the interesting things we found is that measured air channel diameters from the leaves or equivalent air channel diameters, as I explained in the previous slide, do lie within the range that is predicted by the model to give maximal CO2 uptake, which is quite an interesting result. So obviously it's possible that by complicated means, it is a coincidence that our model produces a result that matches with reality, but we're hopeful that the reason that we have a match up here is because the model is effectively recapitulating aspects of how gas exchange works in the leaf. So just for the benefit of those who don't like to think about airspaces in meters, this range is sort of between 10 microns and 100 microns. And the values represented here are from a set of wild type Columbia airspaces. So obviously there is quite a big range that we can see there. So thinking first about just the shape of this graph, this curve, and looking back again at this diagram, there is something quite intuitive here. So what we're seeing is a trade-off between the number and the size of the air channels in terms of what gives you the biggest surface area for CO2 uptake. So obviously the bigger your air channels are within this diagram, the fewer of them you can fit into a given surface area. But equally, the more of them you try and fit in, the smaller they have to be. So your maximum surface area will be an intermediate number of medium-sized air channels. And that really is what is represented by the curve that we see in this modelled relationship. So as well as a number of Columbia plants, we also had a few different mutants from various projects over the years, and we also looked at where their air channel diameters lay. And I'll just overlay that here. So again, they really are within quite a similar range. So what to say about this. So yeah, I think it's interesting that not even mutants which have what visually seem to be quite extreme differences in airspace to the wild type plant are wildly off the mark in terms of that kind of optimal predicted air channel diameter for the CO2 uptake. So one challenge that we found with the model is that while the values for equivalent air channel diameter match up very well with the predicted air channel diameters that we saw in the previous graphs, the corresponding predicted numerical value for CO2 uptake is not quite matched up. So as we saw, our model predicts CO2 uptake, and that parameter, as on the previous graph, is called UNCO2. So that's just saying that that's the uptake of CO2 for a given area called N. And as I explained before, that that parameter is calculated by inputting the CT structural data and by inputting GM, which is calculated from ACI curves. And those same physiological measurements also give us a value for assimilation at various CO2 concentrations, but the equivalent one to the model output should be for an ambient CO2 concentration. So we call that 400 parts per million. So yeah, just to visualize that our issue here is that the model doesn't correctly calculate CO2 uptake. Now, there's quite a number of reasons why this could be, and my understanding, though I'll admit I'm not a modeler, is that it's very common to find these sorts of numerical mismatches in these sorts of models. So I have spent quite some time thinking through the different assumptions underlying the model, whether the likelihood is that they could explain this, or whether they might actually, whether we might expect them to cause the opposite trend in under-predicting assimilation. And I think my overall conclusion, having done lots of poking around and a few extra analyses, is that it's very likely that this mismatch in calculated CO2 assimilation arises from contributions from quite a number of different sources and assumptions. It's very unlikely that just one factor is a big mistake, essentially, that is giving us this this difference between the model and the measured data. But there is something interesting about how this mismatch varies between the different mutant lines we looked at. So what we saw is that different mutant lines vary in the extent of this mismatch in predicted CO2 assimilation. So that tells us that the model is working well for some lines and less well or better for other lines. So this graph is a little complicated to explain, but I'll try and go through it steadily. So since we found this mismatch, we calculated a correction factor, and that was just based on the wild type data. And then when we looked at the mutants, we found that some of them, even when you applied the correction factor, there was still a bit of a mismatch. And so what we did was calculate what we call a deviation factor, which is a multiplier for the correction factor that allows each mutant, or in the case of this graph, each individual plant, allows a perfect match between modelled and measured assimilation. And what we found here, so we looked at whether this deviation factor, which we call alpha, was correlated with any of the structural factors. And what we found was that leaf depth does correlate quite strongly with this deviation factor. So LZ here, I say leaf depth strictly, it's palisade depth, it's just the depth of the palisade mesophyll that's the parameter here. So this might initially sound circular because we but the depth of the palisade is one of the parameters that goes into the model. So what this is telling us is that other factors relating to variation in palisade depth also are driving these differences in how well the model predicts assimilation. So some of the mutants are described very similarly to the wild type, and others, particularly those that differ more from wild type in their palisade depth, are less well predicted. So I guess the next thing for us to really unpack in terms of developing this model is what is it about palisade depth and its variation between mutants that causes it to affect how well the model predicts assimilation. So one obvious possibility is that palisade depth, leaf depth are both related to the light attenuation properties of the leaf, and that is not something that's currently captured by the model. The model is only based currently on CO2 uptake. And as we described right at the beginning of the talk, a fantastic leaf needs to be great at light capture and CO2 uptake, and of course at controlling its water loss. So that's another factor that's not currently built into the model, but that trade-off could be one of the factors that means the model varies in its effectiveness with the depth of the leaf. So we did set out to create a very simplified leaf model and to see whether it could, in its very basic form, replicate some aspects of the real biology of the leaf, the physiology, the gas exchange properties. And yeah, I guess what we saw was that, interestingly, the model does seem to capture some aspects of the structure, but there are some parameters that probably need to be refined or need an additional contribution in the model to allow it to predict things more effectively. So as I mentioned, one thing the model currently doesn't include is effects of light depletion with depth in the leaf. So that's something that we could look to try and add to the model and see if that would help to ensure a more accurate prediction of CO2 uptake. Another limitation of the model in its extremely simple form is that it does only represent the palisade layer of the cell of the leaf. And one potential problem with this is that the spongy mesophyll could also be contributing to the measured CO2 assimilation that we're trying to match up with the model CO2 assimilation, although evidence does suggest that the spongy mesophyll contribution is relatively minor. But that is another area that we could try and think how to represent more effectively in the model. And another factor that I thought is one of the sort of really key obvious mismatches between our simplified leaf and a true leaf, a true Arabidopsis leaf at least, that like the ones that we measured, is that in our model stomata are only on the abaxial side of the leaf, whereas obviously in most Arabidopsis leaves you'll also find adaxial stomata and often about a 2-to-1 abaxial to adaxial ratio. So by assuming that no gas exchange can start from the top of those simplified tubes, we're perhaps missing out on something really critical there. So just to summarise this project and the findings so far and some of the ideas about where to go next, really. So we've generated an extremely simplified model of a leaf using this cylinders packed into a cube visualization. And we have modelled using physical diffusion laws how we might expect CO2 uptake to vary based on the number and the size of the air channels within that area of leaf. We parameterised the model using data generated by a micro-CT approach and gas exchange data, and those data were paired, so we know that they come from exactly the same leaves and they should match up well, so that removes any concerns about differences in growth conditions or other factors which could have affected how the leaf developed its structure and how it performs physiologically. As we saw, the simplified leaf model does predict a maximum CO2 uptake that is a similar equivalent air channel diameter to that we measured from the micro-CT data. So that perhaps suggests that the model is correctly working out that trade-off between the size and the number of air channels that you can fit into a given leaf area. And finally, we have this problem that our model overestimates assimilation, but interestingly, we see that the degree of that overestimation does vary with the depth of the palisade layer, and that perhaps tells us that there are other aspects of the biology that we need to take into account in order to produce a more accurate and effective model that can work across a variety of different mutants with wider variation in various aspects of their leaf structure. So finally, I'd like to thank my colleagues at the University of Sheffield. So Shiv, the PhD student who has done the modelling component of this work, and Professor Andrew Fleming, who was my PhD supervisor and who is also one of Shiv's PhD supervisors, along with our colleagues in the engineering department. And also to thank Dr. Andrew Mathers and Craig Stoick of the University of Nottingham Hounsfield facility, where we were able to do the CT scanning and analysis. I'm currently work at the University of Bristol, but this work was done at the University of Sheffield and at Nottingham, and it was funded by BBSRC, and my work was funded by the Gatsby Charitable Foundation. So thanks ever so much for listening, and I look forward to your questions.
That's great. Thank you very much, Alice. No, complicated indeed, but I think you did a good job explaining some of those concepts. Yeah. So we have a couple of questions. And we have time for a few. So first of all, Andrew Romanowski asks. So he says, "Hey, I really like the CT scan method you use," and he has a question about the model. And does it take into account temperature, or is it at just a fixed temperature?
Yeah, the model does take into account temperature, and the model obviously, so temperature is therefore one of the parameters that we constrained to generate the values that we're comparing. So one of the nice things about having the like or physiological data is that we can control lots of parameters on that machine, including temperature. So when we're comparing modelled and measured outputs, we use the same temperature value in the model as the one that we use to control the gas exchange machine.
Okay, cool. So let me reiterate, if anyone has any questions for Alice, please write them into the question box. So Alice, I want to ask about the mutants. So you were kind of coy about naming those mutants, so we noticed. Yeah. But so let me ask in general though. I mean, you didn't see huge changes, especially in that bell curve that you showed initially. So are these mutants in general that have no stomata or loads of stomata or very minor changes in stomata? Can you give us a bit of an insight?
Yeah, this is a good question, and it was coy for simplicity rather than for anything secret or shady. And they are all public mutants actually. And we used quite a number. So there were a few mutants I was working on that are mutated in various cell wall biochemistry. And there were, I think we included at least one of the EPF mutants that Judy Gray's lab works on. So that's a stomatal density mutant. We also had some of the mutants from the paper I mentioned earlier, where the cell cycle genes were varied. So one of the nice things about having quite a variety of mutants in different aspects of the leaf biology is that it sort of doesn't put all your eggs in one basket, as it were, but equally that makes it a little complicated, because one possibility for why we're seeing mismatches between our modelled and measured values is that there's something specific to different types of mutants. So with the cell wall mutants, for example, you could imagine that GM might be directly affected, and it could actually be to do with the permeability of the cell wall. So that would not be ideal. But I think one of the things that's really interesting and nice about this quite neat correlation we see with palisade depth and our deviation factor is that perhaps suggests that there is something common across the wide range of mutants that we're looking at that that needs adding to the model. So that maybe suggests it's not just sort of some weird cell wall problem or a stomata specific problem. So yeah, does that answer your question?
Yeah, absolutely. No, that's absolutely fine. That's fine. So we got a question from Enrique Lopez, and he asks, so I'd like to decrease chloroplast move in cells away from the vertical walls to the upper and lower surfaces. So chloroplast movement could be part of the reason for overestimating assimilation. Would there be any way of incorporating the possibility of chloroplast movement into this model?
Yeah, so yeah, very much light depletion and chloroplast movement are two of the sort of aspects that we know are big emissions from the model, and I think it would be really valuable to add them in, and I think it is in the plans for Shift's project, though whether he'll be able to complete that before his PhD wraps up, I'm not sure at the minute. Yeah, it is an obvious emission. I think one of the things that's maybe most interesting about the model is while there are lots of big assumptions and emissions, there are some things it does really well, and I think in a sense that's sort of what we hope for in a model, that we can make something very simple that explains a lot. But yeah, I completely agree, it would be really nice to add that kind of light module to the model to look at depletion with depth and chloroplast movement. I guess measuring chloroplast movement, I mean, I guess it's something I do more in my current role than in my PhD work, but it'd be very tricky to get that data in a paired way in any sense, but I don't think it would be impossible. Okay.
Thanks. So we have a question from someone you might know called Andrew Fleming here.
Oh yeah.
So he says, you know, great talk, obviously. So a bit of a question from Alice's PhD supervisor. So with future CO2 levels going up, can the model predict whether the future leaf should have fewer larger diameter palisade cells or shorter air channels in the palisade, or is it too early to say?
My understanding is that the model could predict that, but the current outputs we have from it are all based on an assumption of 400 parts per million CO2. But I think CO2 concentration is an input to it. The only problem would be that we would need measured Arabidopsis data from the mutants at those. Oh, that's not a problem, we have those data. Yeah, yeah, that should be completely feasible actually, because obviously the AC curve data do include elevated CO2 measurements. I suppose what they don't do is look at how growing the plants under elevated CO2 affects their development. So it would give you an instant time point for the given structure, but it wouldn't allow you to assess whether an elevated CO2 would also change the structure. But I suppose in terms of the model, what we're interested in is that fundamental relationship. So it almost doesn't matter how you've reached the structure that you're using. What you're interested in is just how that structure affects the CO2. So yeah, I think the model should be able to answer that question.
Okay. Okay. So final question before we move on to Ansel's talk. So there actually kind of two questions which kind of ask the same thing. One from Ashley and Tapan, and they ask whether air spaces change either in different developmental stages of the plant or under different growth conditions. Yeah. So yes to both. So I'm trying to think of a. So yeah, there are lots of aspects of growth conditions that can affect the way that the airspace is developed. So in a typical Arabidopsis leaf,
a very young leaf has no air spaces, and those air spaces gradually open up over time. There's actually still surprisingly much to learn about how that process happens, but it will be partly driven by turgor pressure kind of pushing the cells to swell and therefore pulling them apart at cell junctions where multiple cells meet one another. And it's also likely that there might be some active cell wall processes involved in unzipping the connection between cells, although that's something we have a really poor handle on at the minute. So yeah, both growth conditions and developmental stage will influence the structure of the leaf. Obviously, for all the measured data here, the developmental stage was consistent and they were leaves that we considered mature. So I guess we would expect most leaves to reach a similar stage ultimately, but I guess there's a whole new can of worms to look at younger leaves as well and how that influences the early development of the plant. But it's an interesting question.
Cool. Okay, thanks, Alice. So we got another couple of questions from Andrew, but I'm going to pass those on to you by email, and you can answer him online. Okay, so thanks very much, Alice, for that.
Thank you.